Hey guys, I'm Jenna and we're going to be doing a video on AOPI staining. If you haven't already watched the cell splitting or the cell counting video also on this YouTube page, I recommend go ahead and watch that before you watch this video, okay?
Hi, I'm Elise, and we just split our cells, so now y'all are going to go count, and then once you come back, I'm going to show you some math to do to dilute your cells to the proper amount for plating. First, you need to determine the number of wells you wish to plate for your experiment, and then you'll multiply that by 1.25 to get the total amount of diluted cell solution you need for plating. This is combined of concentrated cell discard solution that you take from multiplying the number of wells times 10,000, then you divide that by the concentration of cells per mil that you got from counting to get the concentrated cell discard solution you need. Then you take the total diluted amount and you subtract from that the amount of concentrated cell discard solution to get the amount of media you need to add. Then you combine the volume of media calculated and the volume of concentrated cell discard solution in a 50 ml conical tube to get your diluted cell solution for plating. Here's an example from an experiment that we performed in which we used 24 wells for plating and our concentration of cells was 2.34 times 10 to the 6th. We needed 29.897 mils of media and 0.103 mils of cell discard.
Hey guys, I'm Reagan and I'm going to teach y'all how to make dilutions and how to plate. So first, according to our math, we need 29.9 milliliters of media, and then you're going to transfer that into a new conical tube. So to get 29.9 milliliters, I need to use two different serological pipettes. Now that we have the media into the conical tube, according to our math, we need to add 0.1 milliliters of cell solution, and 0.1 milliliters is also equal to 100 microliters. So now that you have the cell solution into your conical tube, you want to close it and mix it up very well. And then once it's mixed, we decided that we were going to put 1.25 milliliters into each well. And since we don't have a serological pipette that's that small and we don't have a pipette that is that large, we decided that we were going to go in twice for each well and pipette in 625 microliters. And whenever you're pipetting into your well plate, make sure that you skip a column of wells so that you don't have any bleed over.
Hi guys, my name is Brie and I'm going to be showing you how to make your AOPI stock solution. So how much you need to make depends on how many wells you're doing and how much you want to put in each well. So we did 20 microliters per well and 24 wells. So that's 480 microliters total of our stock solution. And we're going to make a little bit extra just so we don't run out. So what we're going to do is we're going to take 250 microliters of PBS, and we're using PBS because it's clear and won't interfere with our staining. And then we're going to do 250 microliters of our dye because it needs to be in a 1-to-1 ratio. So what the first thing I'm going to do is I'm going to take 250 microliters of the PBS, 250 microliters microliters. Make sure your pipette is already set. And then add it to the microcentrifuge tube. And then I'm gonna add 250 microliters of the dye into the microcentrifuge tube. The dye is light sensitive, so make sure you do this as quick as possible. There you go, that's your stock solution. And this is what you'll put on your cells.
Hi, I'm Riley, and our cells have incubated for 24 hours, so we're going to take the lid off and we're going to suction the media off of the cells. So we'll plug our stereological pipette into the tubing, and we will suction off the media from the cells, and then when most of the media is gone, you'll lift the plate so that you can get the rest of the media without disrupting the cells. So now that we've suctioned all the media off of the cells, we're going to take our AOPI dye that we previously made and put it along each of the wells. So according to our math, we'll be taking 20 microliters and putting it along the cells. Now that we're done putting the AOPI along the cells, we're going to cover up with a lid and we'll leave it in the dark for 5 minutes, and then after that we'll take it over to the microscope.
Hi guys, I'm Ashley. So now that we're done letting our plate incubate in the dark for 5 minutes, we're now going to visualize our cells with the AOPI stain on them. So first things first, we're going to turn on our microscope. And so to turn it on, you just open up the iPad. Next, a couple pointers before getting into using the microscope. You're gonna have this knob right here. That's going to be for moving your stage up and down. The smaller knob below it will be for moving it side to side. You have this knob right here, which will be coarse and fine focus. So this larger knob will be coarse focus. This smaller knob will be fine focus. And lastly, you're going to want to make sure that your lens is set to 4 times. If necessary, you can set it to 10 times, but we found that 4 times is the best for optimum viewing, and that is this red lens. Something really cool about this microscope is that it can actually be inverted. And so for our purposes, we recommend that the microscope is inverted with the lenses at the bottom. We are also going to be viewing in fluorescence imaging today. So what you're going to do is you're going to take this microscope and on the app switch from bright field, which is this white light, to fluorescence imaging. We recommend that you view your live cells in the phytic filter, which comes across right here. You'll be able to control the amount of light coming through as well as the exposure on these 2 tabs. We recommend viewing your dead cells in Texas red, and it's going to be just that same thing. You're able to control your light right here and your exposure here. And then lastly, we recommend taking a transmission image of your cells, and this will just show you what your cells look like before any fluorescence has been applied to them. So kind of like a control. So now we're ready to visualize our plate. So we've put our plate onto the microscope, making sure that a well is within the visual field of the lens, and we've taken off the cover. So for our purposes, the best thing that we can do is to visualize this using an overlay. So what you're going to do is you're going to go from single to overlay down in the bottom left-hand corner of the microscope. And first you're going to be on phytic, and so this shows you your live cells. You're going to turn on the light. Once everything's in focus, capture the image. And then you're going to move to Texas red and do the exact same thing in the same spot. And so what this does is it generates an image of both the live and the dead cells, so you're able to compare them between each other.
Now you are ready to start visualizing your cells. So we suggest go ahead and take a trans image of your well just to ensure that you know where your cells are located. Then you can go ahead and take the overlay photo that we mentioned in the previous section, and this will give you the picture of live cells that are green and dead cells that are red. This is just another variation of what your overlay can look like, and this is when you can start counting those colored cells to get your viability count. When your overlay looks like this, most likely all of your cells have been killed and there are no live cells in this sample. The way you test this is switching to your Texas Red and seeing that the cells are illuminating and fluorescing red. However, they will show up in the green, but they won't be near as bright. And so this is whenever you can tell that you have killed all of the cells and there are no live cells for your viability assay.